we have to wait for further notice about what will happen about CIA 3 and also the final exams. So what we are going to do today is practicing some of the questions. We have some topics still left. Um, those topics are one of it is NGS, right? And there is one topic I would want to discuss, and there is one more about HIV and CRISPR together. So I will give you a few questions now. I do not have any intention of answering. Rather, I would want you to have some original thinking about it and then see if you can solve it or not. So the important thing is that you should be able to solve. And you can all turn on your mics and discuss openly. Okay? You should assume it is an open class. I hope you do. If there are any physical classes prior to final exams, we can also discuss further more and many more questions. And this is also a good practice or training for for the final examination as well. I'm taking time for this because I would want that you practice as many as possible, so that we all will all be we'll all be able to do much better in the coming exam. So the first one is, you can start with this one. Okay, you can I would like that you try to. I will also post this on the Google Classroom, so everybody has to submit this so that we know how you are doing with this. Can anybody try? Because some people have asked me about we need more practice questions. So this is one of the ways I can help you out with. Can anyone try to address this?
Sir, at the first glance, we can say that person 1 is homozygous, whereas individuals 2 and 3 are heterozygous.
Yeah, 1 second. I'll just complete. Typically, this is the normal one. I didn't mention that. So assume that 1 is normal, and about the rest, you should tell. Yes, continue, please, Parvati.
Sir, a doubt, sir. Like the first one is the first
normal
person is
normal,
homozygous,
and the normal. 2 and 3 are patients. Assume that, patients. Or with mutations. Yes.
Okay, sir.
Yeah. Parvati, you're saying, I guess, I thought.
Yes. Individual 1 is homozygous, whereas
Yes.
2 and 3 are heterozygous.
That's right.
And we can name the alleles for convenience.
Yes. Yeah, you can.
So like we can name as 1, 2, and 3. So first one is homozygous for allele I1. Like I'm just putting this I1, I2, I3. We can put that as homozygous I1, I1. For the second one's I1, I2 heterozygous, and the next one is I2, I3 heterozygous.
Okay. What is the nature of the mutation? That is always the most important thing that we try, is the nature of the mutation. That is what we should try to understand. Okay, for simplicity, we will take this is homozygous for I1, one. Sample 2 is having I1 and it is heterozygous I2. And sample 3 has, this is matching, and it is matching in majority of the cases, so we will call it as I2, and it has a new mutation, so it is I3. Okay, now I would want more number of people to participate in this discussion, and it will be nice that way. Please don't feel anything about whether your answer will be correct or wrong. It doesn't matter, correct or wrong. The first thing is to be able to speak up, to speak and express your opinions. And nobody is judgmental here, and we don't judge anybody. We don't bother if somebody else is judging us. We are interested in ourselves and our growth, and that, and I want that you all be able to speak up, and that will be a nice thing if you can do it. Yes, go ahead. Anybody else? What can you say about northern blotting from northern blotting? So one is having sufficient amount, and there is quite
Like there's no
mRNA.
mutation.
Yes, yes. One is normal, right? So we don't have to bother much about it. We are using this as a control, okay? And we are comparing every 2 and 3 with 1 and see how they are different. If you see in 2, there is no difference in mRNA. You're finding it same amount. So we don't know what it is. And 3, there is something else. But as I said before, there is multiple ways of comparison. You have 3 samples, and there are 2 alleles or 2 genome sets, right? We are deployed in nature. That's what I meant here. And then we have 3 different assays, right? We need to be able to compare all these things and build a mental map and then figure out what is the correct answer. So if I see the 1, 1, and 1, they are normal. So let me compare the second case. It is heterozygous based on southern blotting, that is clearly available, deducible. With from northern blotting to the mRNA wise, there is no difference. So we cannot say much, but at least we can say is it is not a deletion in that particular gene. It's not a deletion. It's not an insertion. We can say that, because if there was an insertion or deletion in that gene, okay, assume we are talking about a gene, it could be many other differences. At least with regard to the gene that we are looking at, it's not a deletion or not an insertion. At least it's not a big deletion or big thing. It could be very small range or something like point mutations. Point mutation could be transition, transversion, that those are substitutions. You can also have single nucleotide deletions or additions also. And because there is no big difference in the mRNA bands, so the gene, it's not deletion and insertion, that much we can infer. Let me see from the Western blot. I see that this is the normal protein size, so this band is there. Although the mRNA is looking like this, is same in size compared to that of the wild type or normal person, you see an increased size here in the molecular weight. What could that cause? The protein is there. It could have produced a protein of this size, but somehow it is producing a larger protein, which would mean that there must have been, first thing is, this protein is mostly is there within this also. We are saying it based on the antibody is recognizing it, so mostly some part at least is the epitope is present in that. And the stop codon might have had a mutation, because of which it might have become a larger protein. That is one of the possible answers for it. But here, if we compare two, it is heterozygous, and now I need to describe what kind of a mutation is here. If it is small, it doesn't mean it is deletion. We don't know it. We cannot infer that it is a deletion. We cannot infer. It could also be an insertion as well, like we last discussed. In southern blotting, we are using restriction digestion, RD I will put it as. So the fragments forming does not necessarily indicate what has happened there. It all indicates about the presence of the restriction endonucleases alone. We are saying it is sometimes it is possible, but it's not conclusive. That's what I meant by it. But with the gene, at least the mutation, what we can identify is it is a point mutation, because the mRNA size is not changing, right? And it could be the in the allele I2, you're having a larger protein, which could be because of larger mRNA. The ribosome is reading further, and that's why you're getting a larger protein, right? Like that. There are also other possibilities. We can never end, but the point is about what are you thinking? How are you justifying your thinking? That's the important part of it. So in sample 3, you see that this is similar to, this is the I2 allele, and we have a fragment that is larger, which would again not mean it is a deletion or insertion. What we know, however, is that there are two alleles, but there is decreased amount of product formed in northern blotting. That means, so there are two alleles, right? I will write the genes here in I2, and I'm putting, and the other one is I3. I2 is forming the mRNA. I3, you don't see any transcription anymore, any other fragments. So probably I3's transcription is not happening. How can we be sure about it? Again, we will have to compare it with the sample 3 here in the protein Western blot. We see a band that is corresponding to that of I2, like we discussed before, but there is no other band that is present here. That means I3 is not transcribed, because of which there is also no protein. So why is I3 not expressed? It could have been a mutation or a loss of the promoter itself. If there is no promoter, I3 is not expressed. Okay. There are a few other questions that I have, similar ones, but it's all about how you want to practice. But we also have to practice many different varieties. I am doing most of the talking. I hope that you will be able to speak more, and that will be better that way. Please try to participate more. I want to keep quiet and let you do it. So I'm giving the next question. The next question is here. I made about 6 questions, and it should be a very good practice for all of us. I will also upload the questions in the Google Classroom if you want to practice. And when you come here, and if there are any physical classes, we will also discuss. But I need you to come up with what you think, and then we can solve. Here, I need to prepare a randomly labeled single-stranded probe. Okay, assume the probe is supposedly the probe is RNA probe for northern blotting for the gene XYZ. And the technique to be used is in vitro transcription, and this is the vector that is given here. What should I do? Tell me in 3 or 4 steps, and that should be the answer for it. What I need RNA molecule, and it should have randomly radio labeled. How can we get that? Anybody, please try. Is anybody answering? So if this, I am trying to do northern blotting. what is northern blotting? It is trying to detect the RNA, or in this case mRNA. What is the mRNA? This is DNA, and if it is transcribed, say ATG, da 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 da, you will have and a stop codon, that is TGA. And when we were describing about it, we described about how do we call these strands, right? If mRNA, if this gene is transcribed by RNA polymerase, mRNA will be produced. mRNA would be having the AUG and UGA. This is the open reading frame, right? Now I need to detect this one. First thing, this mRNA is called as plus strand because it has the open reading frame. It is also called the coding strand. It is also called the sense strand, and so on. If I need to detect it, I need to have something that is complementary to this, to the mRNA here. So that means I need to prepare the antisense strand. We have the given way, there is a vector. It has the gene that is cloned, that is XYZ. And there are two promoters of T7 polymerase here and here. If I add if I have to do in vitro transcription, I would have added T7 polymerase, T7 RNA polymerase. I would have added ribonucleotides, triphosphates. I should also add CTP or any of the bases, one of the bases that is labeled, that is radio labeled with P32. And which phosphate? There are three phosphates here, right? Phosphate, and this will be the alpha, beta, and gamma. We don't want to overuse radioactivity and release the radio elements out into the nature. It's dangerous. So we want to limit the amount of use. So we have to choose only one of the bases which should be radio labeled. And because this is the alpha phosphate is the only one that is going to get incorporated into the growing nucleotide sequence, we would have chosen alpha P32 labeled CTP along with also NTP. And we need to add the template. If I add this vector that is given here as template, using this RNA polymerase, one strand will be produced. And there is also another polymerase, T7 polymerase here. So the second strand will also be produced. These two are complementary and they will bind like this. And we cannot use this as a probe because they are complementary and they will base pair. So we need to do something else. We need to do something so that we can get, we as I said, we only want the antisense strand. We don't want the sense strand because we have to recognize or identify or say base pair with the mRNA. This is the one that we want to detect. So the idea is simple. We don't want two strands, so we can use either cleave it either with EcoR1 or BAMH1, so that we will get only single stranded, right? Because if we cleave, say for example, if I cleave it here, this RNA polymerase, this promoter will allow the production of the mRNA, whereas this T7 polymerase will only produce this much. It will not produce the transcript of the gene itself, right? So what we can then do is, but the problem is, it is a single strand. Is it sense strand or antisense strand? It is a sense strand.
Sense strands.
That's right. So this is not what I should do. Shouldn't be using BAMH1 to cleave it. Rather, I should use EcoR1 to cleave it. Now the promoter here will only allow the transcription till here on the sense strand, but it's not producing the whole mRNA. Whereas this promoter that is on the downstream, when it transcribes, it will produce the antisense strand. And that antisense strand could be used as probe. And it is radio labeled. I mean, when you cut it and allow the in vitro transcription, you will get the required probe. So I hope you see that we are in a level of higher thinking. It's not just learning about how to construct just a vector and those things. At least 50% of the paper, you may have some questions like these. And that is what is analytical thinking. That is what is research aptitude, right? Okay, let's try one more question. But I was expecting that you will do most of the answering, and I am ending up doing it. So the next question is simple, and I want you to please try to do it by yourselves. Here is a question that is given to you, and what you are supposed to do is two samples are given. One of it is one allele. Assume that it is one allele, and this is second allele or a mutation. Say this is one bacterium, and this is a same bacterium or a mutant of it. And you have sequenced it, and you need to. I guess you can see. Is it Maxim Gilbert method, Sanger's method, or NGS? What
Sanger,
kind
sir.
of it
Sanger.
is Sanger's methods. That's right. And so you should know how to read it, read the sequence and write it in one way. And for sample 1 you write, and for sample 2 also you write, and then see what is the difference between them, and see what kind of mutation it is and what are the consequences of that mutation, right? This is able to determine the sequence is the technical part, and this is important as a biologist, as a scientist. If we know only this much, we are probably technician. And if we know to see more, to get more information in a qualified way, that is when we are scientists. We are thinking like scientists, or we are able to performing like scientists. So can anybody help me out in sequencing? What would this be? And Sanger's method, how would you do? You would take the template. You would have a primer, right? I will, for ease of ease, I will just use a single strand and just recollecting the promoter. So this is 5 prime to, this is the template, and we have added a primer 5 prime to 3 prime, and we would have added 4 different ATPs, right? It is also called as dideoxy method because we use dideoxy nucleotides. And yeah, we will have 4 different reactions. One is with A, one is with T, one is with G, and one is C. In each of them, we would have added at least one dideoxy. Here, dideoxy ATP, dideoxy TTP, GTP, and CTP. And then we will get different fragments, and each of them we will run. And this is the sample. This is the representative gel. Now I need to read the sequence. The difference between Maxim Gilbert method and Sanger's method is that we have a sequence like this. When what we see on the gel is the sequence of the bottom strand here, right? It is the sequence of the bottom strand. If the question were to say, what is the sequence of the top strand, we should have made the reverse complement of it. Only then we will get the actual sequence. So can you tell me, first let's fill up this, what is the sequence here? Can you tell me what would this be? This is T, I'm reading this one.
Sir,
Okay. Yeah,
TCA,
yeah, A,
TCT,
TCT,
GAT,
GAT,
CC,
yeah,
GCT,
this is T, and then G, T, A,
GC,
C,
GT.
A, T. Is that correct? Anybody has any?
I got this, sir.
Yeah, okay. The next one, can you please also?
I'm doing it, sir.
I would also want other people to speak as well, please. Somebody else can tell the for the other one.
So TCAA,
One second. From bottom we are going. Yes,
yes sir.
T C A A
TCT,
T C T. I'm not looking. You only have to take responsibility. G A T.
ATC,
Say, uh,
ACGC,
C T C G C. Yes. Are we? Is
there
there
will be
any
an
different
A, sir.
here?
GATC, yes
One
sir.
second. One second. Let me go. Where I should I? This is fine. And here
GAT,
is C A. Uh,
C,
yes, C G
GCT,
C T
GTA,
G T A
CAT.
C A T. Okay, that's correct. So we now need, this is the sequence of the bottom strand here. I want the sequence of this one. So we should actually, so we should also remember that we are writing this as 5 prime to 3 prime in both the cases. So let us make reverse of it. I will try to write if you can tell me very accurately. Yes, tell me A. I'm talking about this sequence. Tell me the reverse complement of it. And it would be, one second. Sorry. Yeah, I want a reverse of it. So I have to actually write A. That would be the 3 prime. A. Next. A, G,
TT,
A, G, T, T, A, G, A, C, T, A, G, G, A, sorry, C, this is C, right? Yeah, G, A, C, A, T, G. Okay, tell me if there are any mistakes, but otherwise, can you read the sequence now? A, G, next, T, T, A. Come on, keep telling me, give me some, I need help. A, G,
AC,
right? Yes, A, C, T, next,
TA, GGCG,
A, G, G, C, G,
CG, AC,
yes,
AT,
A, C, A, T,
GTA.
okay, yes. Now we have to do for the bottom strand. A, keep doing on your paper. It will be nice if you can do it along with me, because this is how the answers would be expected, something like this. So what is this? Is CG? Yes. Okay. Now I have to write the sequence here too. You can help me. A, sorry,
GTT,
A, G, T, T,
AGA,
A, A, G, A,
CTAG,
C, T, A, G,
TGCG,
T, G, C, G,
ACAT,
A, C, A, T,
GTA.
G, T, A. Now, can you describe? Okay, we got to the sequence. We determined the sequence of the template. Okay, you have to read the question here. I only wrote a few sentences. Usually, the question will describe whatever I describe now. The sample is given to you. 2 samples. One is mutant. One is normal. How would you you performed this? You got the representative gel, then determine the sequence, and then describe about the mutation. What can you tell of the mutation?
It's an insertion, sir.
Where is the insertion?
12th,
The 11th position.
11.
1, 2, 3. You have to also look more, right? 3, 4, 5, 6, 7. This is the stop codon. This is the start codon. So 1, 2, 3, 4. 1 second. Did I have anything wrong? 1, 2, 3, 4. Let me see. Yeah, let's go ahead with this. 4 and 5, 6, 7. Is that correct? Or there should be a mistake somewhere. Did I miss a base anywhere? This question was made long time ago, the diagram. So, okay, anyway. So what can you tell of the mutation here? It is an insertion, that is right. It is a point mutation and it is an insertion. And then, yes, the consequences should be described also at the level of protein. First thing is you need to identify it is an open reading frame. You have to look for open reading frame. Open reading frame would have a start codon and a stop codon with continuous codons like this. If you didn't even get the idea that you have to look for open reading frame, then you are not looking at the hard enough at the data that is given to you. So look at if there are any open reading frames, if you find them, and then you have a different view about what the consequences of this insertion can be. And what happens into the open reading frame in the second one is there is an insertion because of which there is a frame shift, right? So you need to look at the data as a scientist and trying to squeeze out as much information as possible in the given sequence, given question, okay, or data. I hope you like this question. One of the questions did come for CSCR as well, something like this. Okay, we have 20 more minutes. I will give you a few more questions. Give me a minute. Okay. Yes, some of the questions like this are also likely to come. We will do 2 questions at least. This is a simple one. Let's see how you will perform it. You must have learned this before in some others. So the idea is I have a gene X. Okay, there is a gene X. I need to clone it, and I want to get this protein. This is a novel one, so we don't know anything about it. My objective is to have a large quantity of protein so that I can sequence or I can characterize it. Okay, but all I have is I assume that we have primers for gene X. That's it. We don't know anything else. What will you do? Or what will be the steps to make this? And I also gave you a condition or a requirement. I want to have a large quantity of this protein so that I can perform different types of tests for gel, for size determination, for sequencing, to see if it has any enzymatic activities and many other things. So I want to use nickel-based immobilized metal affinity chromatography. So what should I do as a recombinant DNA technologist? How can I achieve this goal of getting large quantities of the protein using nickel-based system, purification system, affinity chromatography, right? Yes, anybody?
So we should could clone it in an expression vector, and maybe when we are amplifying the DNA, we can add the code for the histidine amino acids so that we will be able to isolate the protein
Okay,
as well.
we clone the gene X under a promoter. What promoter would you suggest? We have to talk specific details.
So if it's an eukaryotic protein, we can go
No,
for
it
gas.
is a eukaryotic protein, but we do majority of the things in bacteria. Say, I am talking about E. coli. So what would you expect? Come on.
Lac promoter, sir.
That's right. Okay, you can use lac promoter. There are arabinose promoters. There are many different types of promoters. We should learn about this. If you use lac promoter, what should you use as an inducer?
IPTG.
That's right. You can use IPTG. And I should have constructed with the lac promoter so that it's highly induced. And gene X is present. And then what should I do? What else?
So
Yes.
since it says the protein has to be purified using nickel-based metal
Yes.
affinity chromatography, we'll have to add
Mm.
histidine tag to it.
What is histidine tag?
6 to 7 repeated units of his amino acids, sir.
That's right. You have 6 codons. You have to add 6 codons of histidine. When you are now you want to do that. First of you have a gene, right? And you have to amplify with primers. And primers, you can incorporate the his tag in the primer itself, right? And then you can clone it the whole. So when it is produced, you will have protein X with 6 histidines here, which can then be used using nickel-based immobilized metal affinity chromatography. There it will have nickel. This his tag will have has high affinity for nickel, and it will all those proteins with the nickel will be retained here. The rest of the things will be washed out. How do we elute in this column? How do we elute? You
We
have
use
studied this in
imidazole.
purification, right? Gel purification, downstream processing, and many other things you might have read. Yes, what you said is correct. Say it again.
So we use imidazole and change the pH
Why do you use imidazole?
because it has higher affinity when compared to the histidine tag that we might be using. So finally,
Why? Okay, this is just a question. The way I learned is important. That I wanted to say this is the question when I was learning about it. That histidine has a side group and it is imidazole ring, right? It is binding to nickel. So we just put in more of imidazole. Okay, so this imidazole ring of histidine is outcompeted, and this one will fall off. Histidine along with the protein. Is that understood?
yes, sir.
Histidine has a side group that is imidazole. And of course, pH changes can also be used to reduce the interactions so that we will get this. So that is one way of doing things. That's right. This is, we spoke about only one histidine tag, but there are many other types of tags as well. I will upload a document. I want you to look at the diagram and be able to write some things. Okay, the last question for today. Okay, not last question. We'll see if we can squeeze more. Yes, I want you to design a promote vector, and I'm giving you certain situations and see if you can design the vector. Let's see. You are the technologist or scientist, and these are the conditions that are given. You are supposed to design a vector, and the vector should be useful to be manipulated in E. coli cells. Um, Okay, we want it should be an expression vector. Okay, we forget about this. We want the expression to happen in heart cells of mice. Okay, manipulations we are doing here, and expression we want it to, that means it is a shuttle vector. You can think of it like that, where you can use it in E. coli as well as in mice. So if and the condition is given to you where the gene should be expressed only in heart cells of mice, not all cells of mice, what would you do for such plasmid? These are theoretical questions. It's about practicing how to think for rDNA. Yes, anybody, please. I would also want more others also to speak.
So there is an answer in the chat box.
Okay, they couldn't. Let me see. Okay, that is a good start. Yes, we will go with first thing is origin of replication. And that should be for two organisms. One is E. coli, right? And we are also planning to say express, this is a theoretical question, we'll just put it as such. So we should have had an original replication that could be recognized in mice as well. Okay, assume just for sake of understanding. And the Yogeshwaran also said that is correct answer. We have to put the gene, and the promoter should be tissue specific. At least what we can say with the heart, we can say myocardial protein, right? That makes up the myocardial muscle. We can use that one, at least we can say like that, right? So we have to put that promoter, and there are also other things we need. We should have a selectable marker. We should have marker that should be selected in E. coli, and also that should be selectable in mice for it. Okay. Yes. So people, the questions could come like this, and you are supposed to think beyond simple just making this. I give everything, and then you describing is different. I give you conditions, and you designing is different. That needs more level of free and open thinking, right? If I give you all the details and tell you to write, then you will say, you will probably identify what it is, and you would also probably be able to say why is it there or its significance. What if I gave you an empty one like this and told you to build? Can you think of what are all required and then put? Here you are given, you are left out in the open, and you are supposed to think and come. So these are two whole new levels. And when it comes to designing, that is the or creating, that is the highest form of thinking. So I would want you to practice these more so that you will feel confident in your future careers. The last question is simple. Yeah, well, I would want you to also use to think and come up with designing an experiment. It is also part of our actual syllabus, which is mentioned. Isolation and sequencing of all the promoter regions in a bacterium. So say bacterial genome is like this, and it has several genes, and each of them have promoters, right? Now I have a very ambitious idea that I want to isolate all the promoters or majority of the promoters. What can I do? Think of any possibility about how you can probably achieve this task. Anybody?
So normally there'll be different types of promoters. So if we are sure
Yes.
about the type of
This is
bacterium and the
we'll
pH.
talk about. We'll talk about. Yeah, it is bacterium. Yes. Go ahead.
So if we are sure, we can start with the -30 and the other regions which we already
No, you don't
know.
have any sequence. You don't have any sequence of it. You don't have the sequence of the genome. No.
So we could build a genomic library.
Okay. Then what do we do? We'll make a genomic library. Then what should I do? That step is correct.
Inside the genes and look
I don't know if it is your network or my network. Somebody's is your voice is cutting. Can you breaking? Can you try again, please?
we can identify the genes and look at the upstream sequence
Is
of
your
it.
network good or is it my network? Can
Her
anybody
voice
say
is breaking, sir.
mine or hers?
Parvati's voice.
Yeah, Parvati, can you check your network once? Please do continue with saying though. Prashanti Lalita, are you there? Can you try answering? And that's all right. Don't worry about the network issues. That's okay. Prashanti, Sundararajan, Srividya, Ramya, Prahladan. Yes, anybody, please speak. If I say the answer, I am dropping you off the opportunity to think by yourselves. The objective of this whole thing is not getting the, it's not about getting the answer, but it is about thinking. Okay, Yogeshwaran, please go ahead.
So open reading frame starts with start codon and stop codon. So we know that codons,
But we
considering
don't
that,
have the sequence.
okay,
We don't have the genome sequence of it. That's why we are trying to find out. Okay. At least one of the experiments is this way. If it is given like this, how would we approach it? So I will try then. So the idea, at least in one of the proposed in the book also, you can find it. There is a vector, and in the vector, they put in a reporter, say for example, like Z. Okay, and it has origin of replication, selectable marker, everything else. And here is the this gene does not have any promoter. No promoter is present. Instead, they made the cloning site here. Okay, this is the cloning site. After making genomic library, we'll have several different types of fragments, right? Some of them with open reading frames, some of them without open reading frames, some promoters, some regulatory elements, silencers, any of these things. When you make a library, genomic library, when you're making, you make this, you would have isolated DNA, fragmentation, and then clone it here. And after cloning it into, or ligate it into this vector, use this as the reporter vector, you can call it as, and then put it into bacterium. So lags it and put it in the bacterium and grow it in xgal. Okay, and this bacterium should be laxate minus. Okay, now there are two possibilities. Majority of these cells, because sorry, yeah, some of them will be blue in color like this, and some will be white. Why would some be white and why would some be blue? Yes, anybody?
Complementation.
Yeah, but
Alpha complementation, sir.
that is all right. That's not why I'm asking. Say if I have the bacterium with which is laxate minus, okay, in this plate, on this plate I only added the bacterium with laxate minus, which is laxate minus. I did not add any vector. What color would you expect the colonies to be? Blue color or white color?
White, sir.
Why
Black
is
is in
that?
minus means white, sir.
Why?
Because lag Z will not be transcribed and galactosidase will not be there, so that the XGAL will not be
Motion won't take
converted to
place.
produce a chromogen.
That's right. So here we are going with xgal and ampicillin resistance. Assume this is the ampicillin resistance. So why are some blue and why are some white? Why could they be? What could have happened to them that they are blue?
If the cloned fragment is a promoter, then lag Z would get transcribed and
Yes,
therefore the further steps.
that's right. So in this cloning site, where some of them may get promoters, some of them may get open reading frames, some of them other regulatory regions. Which regions of those will allow the transcription of this would be if a promoter happens to be cloned here, then or ligated here, then the laxate would be expressed and it will convert the xgal into a blue compound and we will get blue colonies. So then we pick them up and we go for sequencing and try to identify that sequence. Okay. This is not a perfect system, but at least we will get many sequences or promoter sequences. I hope I hope you understood a lot of these, and I hope you have enjoyed the questions too. I would like that you practice little more. And to end this, I will also upload this one in your the Google Classroom. Try to solve it and try to make as complete an answer possible. That's very important, making a complete answer. So this will be the last one. Don't just excuse me for, yeah, last 2 minutes. All I need is to critique this DNA construct. Somebody is making a transgenic animal. Their objective is they had gene X and gene Y, and here they need to integrate. The objective is to put in gene Z, and to achieve this knock-in, okay, knock-in means putting in the gene, they have designed a DNA construct like this. So tell me what will happen if we use this.
So just a small doubt. Does
Yes.
X and X dash and Y and Y dash mean the same thing?
Yes, assume it is Y, same thing. Yes. This is a skill for critiquing. You look into some data and then you say, or you're troubleshooting, something has not worked out. And you need to look at different things and see where the problem is and try to rectify that problem. Right? You don't know if when we really do in the lab, many things will go wrong. Here I am pointing you already that the mistake is in the DNA construct. Find the mistakes. Now you have to find the mistakes in that. It is a faulty one. So tell me what is wrong about it. What are all the things that are wrong about this? I have told you the objective. Objective is to knock in gene Z between X and Y and nowhere else. So now you can comment. First thing, I hope you identify. See
And the open
this.
reading frames.
What is NPT?
Neomycin phosphotransferase gene.
That's right. It's a neomycin resistance gene. What are those arrowheads?
Is it promoters?
No, they're not promoters. Promoters usually we represent it this
So is
way.
it an open reading frame?
No, these are the open reading frames. Yes. Parvati, you're saying?
So the sites of recombination.
That's right. Recombination, recognition sites, right? Or, yeah. Hey, say LOXP. That is one type of recognition site. And it is recognized by Cree recombinase. So what do we need to remember about it? Where should they be placed? I think I will leave it as an open question. Don't want to answer it. But I hope you understood the question. GeneX, the objective is understood. We need this. What is TK? Why is it there? What is TK? Anybody?
Is that a transcriptome kinase?
No. Try a little more. Kinase is correct.
Thymidine kinase.
That's right. It is thymidine kinase. What is it used for? Any idea? What is NPT for? NPT is for
Selectable work
Confirmation.
positive selection, right? For all those that have got this DNA. Out of all those that got this DNA, we have two possibilities. One is at the target site like this, and we may have non-homologously integrated DNA, right? So I will leave it this. I want you to watch the transgenic animals videos once again, have a look at it, and then try to answer this. Okay? I think I
Options.
okay. I gave you why there are two reasons for why I took this class now. I actually uploaded an assignment where I want you to make questions. Okay? And imagine if all 30 people made 4 questions each, we'll have a big repository of questions that we could all practice on that. And to give you an idea, I have taken this class today with trying to give some examples on this, and you can try those things too. And it'll be even better, or actually that is the full exercise is make 4 questions and make 4 answers. Answers need not be complete written stories. I want steps like what we have discussed right now. This is just for practicing how to think like with a person who is setting the questions, and also providing what you expect as an answer, and try to make these analytical questions. Then you are twice benefited. And if you can do this all, it will be a great service to everybody, including me. I will also know your questions, and maybe you will win, and maybe I may not. Yes? Okay, take care, and I hope I'm expecting very active participation in terms of that assignment. Please do that well. Okay? Thank you.
Yes, sir. Thank you,
Yeah,
sir.
Just
take
a
care.
small request,
Yes,
sir.
please. Yes.
So shall we have the deadline as Saturday or Sunday so that we would be able to invest more time and do it
No, more
better?
time is not going to have any benefits out of it. When is Friday I gave, right?
Yes, sir.
Today is Wednesday. I gave it yesterday, right? Question. Did you prepare one question?
Thinking about
Yes.
it, sir.
Okay, I'll make it Saturday, but usually